Good morning, students. Let us continue with the chapter number 14, respiration in plants. Let us start the process of glycolysis, in which glucose gets break down into two molecules of pyruvic. The term glycolysis has originated from the Greek word glykos, that means sugar, and lysis means splitting. The scheme of glycolysis was given by Gustav Ambeden, Otto Meyerhoff, and J. Parnas, and is often referred to as EMP pathway, Ambeden-Meyerhoff-Parnas pathway. In anaerobic organisms, it is the only process in respiration. Glycolysis occurs in the cytoplasm of the cell and is present in all living organisms. In this process, glucose undergoes partial oxidation to form two molecules of pyruvic acid. In plants, this glucose is derived from sucrose, which is the end product of photosynthesis, or from the storage in form of starch. Sucrose is converted into glucose and fructose by enzyme invertase, and these two monosaccharides readily enter the glycolytic pathway. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of enzyme hexokinase. This phosphorylated form of glucose then isomerizes to produce fructose 6-phosphate. Subsequent steps of metabolism of glucose and fructose are same. The various steps of glycolysis are depicted in figure. In glycolysis, a chain of 10 reactions under the control of different 10 enzymes takes place to produce pyruvate from the glucose. While studying the steps of glycolysis, we have to note the steps at which the utilization or synthesis of ATP or NADH takes place. So, at which step which enzyme is involved and what is synthesized or utilized, we have to remember. ATP is utilized at two steps. This we have to learn first at the step when the glucose is converted into glucose 6-phosphate. This phosphate comes from ATP. And second, in the conversion of fructose 6-phosphate to fructose 1,6-biphosphate. Again, the phosphate comes from ATP. So the fructose 1,6-biphosphate split into dihydroxyacetone phosphate and 3-phosphoglyceraldehyde, that is PGAL. We find that when PGAL is converted to 1,3-biphosphoglycerate BPGA, one molecule of NADH is utilized and forms NAD+. Two redox equivalents are removed from PGAL and transferred to the molecule of NAD+. PGAL is oxidized and with inorganic phosphate to get converted into BPGA. The conversion of BPGA to 3-phosphoglyceric acid, that is PGA, is also an energy yielding process. This energy is trapped by the formation of ATP. Another ATP is synthesized during the conversion of PEP into pyruvic acid. So, whatever the number of ATP are utilized, same are synthesized, but when they were utilized, they were the single molecules, but when they are synthesized, they get doubled. Pyruvic acid is then the key product of glycolysis. Fate of glycolysis depends on the cell need. Here, let us see glucose 6-carbon compound utilizes ATP and becomes glucose 6-phosphate in the presence of hexokinase. Then it isomerizes to fructose 6-phosphate. Then 1,6-biphosphate. Again, utilization of ATP is there. After that, two molecules are synthesized, triose phosphate and triose phosphate. Both forms are different, out of which PGAL converts into BPGA. Then again, if this BPGA converts into PGA, one ATP is synthesized, after which it forms glycerate and then pyruvate, where water is released. Now when this phosphoenol pyruvate, that is PEP, is converted to pyruvic acid, again two molecules of ATP are synthesized. So when they are utilizing, they are single molecules, and when they are synthesizing, they get doubled. There are three major ways in which different cells handle pyruvic acid produced by the glycolysis. These are lactic acid fermentation, alcoholic fermentation, and aerobic respiration. Fermentation takes place under anaerobic condition in many prokaryotes and unicellular eukaryotes. For example, oxidation of glucose to carbon dioxide and water. However, organisms adopt Krebs cycle, which is known as aerobic respiration. This requires oxygen supply. So, fate of the pyruvic acid depends upon the cell. If the cell is anaerobic organism, or if it is a prokaryote or unicellular eukaryote, then it will undergo the anaerobic respiration like fermentation, either lactic acid or alcoholic. But if it is aerobic, then it will undergo Krebs cycle, that is known as aerobic respiration. For aerobic respiration to take place within the mitochondria, the final product of glycolysis, pyruvate, is transported from cytoplasm to the mitochondria. Yes, because we must know that glycolysis occurs in cytoplasm. So whatever may be the product will be produced in the cytoplasm. We have to transfer this from cytoplasm to the mitochondria because aerobic respiration takes place inside the mitochondria. The crucial event in aerobic respiration are the complete oxidation of pyruvate by stepwise removal of all hydrogen atom, leaving 3 molecules of carbon dioxide. Next is the passing on of the electrons removed as a part of hydrogen atom to molecular oxygen with simultaneous synthesis of ATP. What is interesting to note that first process takes place in the matrix of the mitochondria, while the second process is located on the inner membrane of the mitochondria. Pyruvate, which is formed by the glycolytic catabolism of carbohydrate in the cytosol after it enters the mitochondrial matrix, undergoes oxidative decarboxylation by a complex set of reactions catalyzed by pyruvic dehydrogenase. The reactions catalyzed by pyruvic dehydrogenase are as follows. Here pyruvic acid, coenzyme A and NAD plus are required. In the presence of magnesium ion and pyruvic dehydrogenase forms acetyl coenzyme A, carbon dioxide, NADH and water. During the process, 2 molecules of NADH are produced from the metabolism of 2 molecules of pyruvic acid produced from 1 molecule of glucose. Hence, we are counting it twice. The acetyl coenzyme A then enters a cyclic pathway known as tricarboxylic acid cycle, more commonly called as Krebs cycle because it is named after the scientist Hans Krebs who first elucidated it. Elucidate karna matlab find out karna. So TCA, tricarboxylic acid cycle, also known as Krebs cycle, starts with the condensation of acetyl group with oxaloacetic acid, acetyl group of acetyl coenzyme A and OAA and water to yield citric acid. The reaction is catalyzed by the enzyme citrate synthase and a molecule of coenzyme A is released which was utilized during the first reaction. Citrate is then isomerized to isocitrate. It is followed by 2 successive steps of decarboxylation leading to the formation of alpha ketoglutaric acid and then succinyl coenzyme A. In the remaining steps of citric acid cycle, succinyl coenzyme A is oxidized to OAA, allowing the cycle to continue. During the conversion of succinyl coenzyme A to succinic acid, a molecule of GTP is synthesized. Here, the nitrogenous base is guanine. This is a substrate level phosphorylation. In a coupled reaction, GTP is converted into GDP and the simultaneous synthesis of ATP from ADP. Also, there are 3 points in the cycle where NAD plus is reduced to NADH and 1 point where FAD plus is reduced to FADH2. The continued oxidation of acetyl coenzyme A via the TCA cycle requires the continued replenishment of oxaloacetic acid, the first member of the cycle. In addition, it also requires regeneration of NAD plus and FAD plus. The summary equation of this is written as follows. We can see that pyruvic acid plus 4 NAD plus FAD plus twice H2O plus ADP plus inorganic phosphate in the mitochondrial matrix gets converted into 3 molecules of carbon dioxide, 4 NADH, 4 H2O, FADH2 and ATPs. We have till now seen that the glucose has been broken down to release carbon dioxide and 8 molecules of NADH plus H plus 2 of FADH2 have been synthesized besides just 2 molecules of ATP in TCA cycle. You may be wondering why we have been discussing respiration at all. Neither O2 has come into the picture nor the promised large number of ATP has yet been synthesized. Also, what is the role of NADH and FADH2 that is synthesized? Let us now understand the role of oxygen in respiration and how ATP is synthesized. Let us have a look on the Krebs cycle. Pyruvate converted into acetyl coenzyme A, converted into citric acid, converted into alpha ketoglutaric acid, converted into succinic acid, then malic acid, and then OAA is replenished. This way we can see how the NADH are synthesized, GTP is synthesized, and FADH2 are synthesized. In the first step, how the one carbon dioxide molecule is released, second and third also. This is the TCA cycle, Krebs cycle, that occurs in the aerobic respiration.